Hi, my name is Pamela Coons, Associate Professor of Medicine in the Division of Oncology at Yale School of Medicine and Yale Cancer Center. I'm excited to announce ASCO's new open access journal, JCO Oncology Advances. As the inaugural editor-in-chief, I hope to support JCO Oncology Advances to become the premier platform to bridge the gap between accessible scientific research and clinical care. Stay tuned for more information, including new article types, at ascopubs.org/JCO Oncology Advances. We look forward to seeing your submissions in spring of 2024.
This JCO podcast provides observations and commentary on the JCO article Results of an International Randomized Phase 3 Trial of the mTOR inhibitor Ridaforolimus versus placebo to control metastatic sarcomas in patients following benefit from prior chemotherapy by Demetri and colleagues. My name is George Demetri and I'm a medical oncologist at the Cowan Cancer Centre and Royal Melbourne Hospital in Melbourne. My oncological specialty is medical oncology with a subspecialty focus on sarcomas. The manuscript presents the results of a large international randomized double-blinded phase 3 clinical trial which sought to compare the efficacy of the mTOR inhibitor Ridaforolimus to placebo by randomizing patients with metastatic soft tissue sarcomas who were benefiting from cytotoxic chemotherapy, which was defined as stable disease or a RECIST-defined response to either Ridaforolimus or a matching placebo. This was a somewhat novel approach, at least for soft tissue sarcomas, as the trial was designed to demonstrate that Ridaforolimus could serve as an effective maintenance therapy following but prior to the development of resistance to cytotoxic chemotherapy. Why Ridaforolimus? By way of background, signaling through mammalian target of rapamycin or mTOR pathway has been shown to be dysregulated in most soft tissue sarcoma subtypes. Ridaforolimus was previously tested in a phase 2 trial in patients with advanced sarcomas and showed some degree of clinical activity with durable stable disease. These results both served as the rationale for the conduct of this phase 3 study, which was called a SUCCEED trial, phase 3 sarcoma multicenter clinical evaluation of the efficacy of Ridaforolimus, as well as influencing the study design and primary endpoint, a prolongation of progression-free survival in this maintenance setting. The primary endpoint of the study was met with a statistically significant improvement in progression-free survival, which was 17.7 weeks for patients treated with Ridaforolimus compared to 14.6 weeks for placebo. With a total of 711 patients enrolled, the hazard ratio was 0.72 and p-value of 0.0001. There were other clear signals of biological activity for Ridaforolimus in these patients, as evidenced by a significantly higher clinical benefit rate, 40% versus 29%, and a mean decrease of 1.3% as the best response in target lesions in the Ridaforolimus group compared to a 10.3% increase in the placebo group. How do we interpret these results in a clinical context? And therefore, what impact will these results likely have on clinical practice? Firstly, although there was a significant improvement in progression-free survival with a 28% reduction in risk in the actively treated group, this only translated to a 3-week difference. As the authors correctly state, this improvement can only be considered modest at best, particularly when taking into account the toxicities associated with taking Ridaforolimus compared to no active therapy. The disconnect between the highly statistically significant result with only a modest clinical benefit can best be explained by the fact that the assumptions on what the progression-free survival of the control or placebo group would be were markedly overestimated. It was estimated that these patients would take approximately 6 to 9 months to progress, but in fact this proved to only be about half of that estimate at 14.6 weeks, or approximately 3.5 months. This is an important lesson to learn, as clearly a better estimate of progression-free survival would have allowed for a smaller study that was not as overpowered as has become apparent from the Succeed trial. Additionally, this modest benefit needs to be balanced with the toxicity of taking Ridaforolimus. Although reasonably tolerated, expected toxicities from this mTOR inhibitor were common, with severe or grade 3 adverse events seen in 64% of patients compared to 25% in the placebo group. When considering that Ridaforolimus is being used as a mechanism of delaying progression, and with this a hope that disease-related quality of life could be maintained for longer, the toxicities of this agent would need to be carefully taken into consideration. The results of this trial are therefore unlikely to have a major impact on clinical practice, with regulatory agencies such as the FDA rejecting approval of Ridaforolimus for this indication. However, it would be disappointing if this class of agents was dismissed as a potential therapy for certain sarcoma subtypes, given the clear biological effects seen in this study. Efforts should therefore continue to focus on exploring rational combination approaches, and in parallel with attempts to better define subpopulations of patients with tumours that are more or less dependent on signalling through the mTOR or other related pathways. The investigators in the study have collected tumour samples from treated patients, with analyses ongoing on identifying potential predictive markers to mTOR inhibition. These results will, of course, be eagerly awaited, but it remains to be seen whether the molecular tools available to us at this stage are sophisticated enough to allow us to determine this with any confidence. Finally, the investigators must be commended for successfully completing this very large international collaborative trial as we continue to refine sarcoma subpopulations further and hopefully define markers that may ultimately predict response to a particular agent, having the ability to translate those findings into practice through international trials, including those with niche patient subpopulations with novel study designs, as demonstrated here, is critically important. This concludes this JCO podcast. Thank you for listening.
For more original research, editorials, and review articles, please visit us online at jco.org. This production is copyrighted to the American Society of Clinical Oncology. Thank you for listening.